Hi, everyone. My name is Kasia Proctor, and I'm going to be your host today. This is a two-part webinar. We will hold a Q&A session at the end of each parts. If you think of questions you'd like to submit, you can submit them at the bottom of the Q&A chat. The presentations will be recorded, and we will be sending out the links a couple days after the recordings. So Max is our speaker today. Max is a field application scientist here at Biolog. He holds his PhD in microbiology from University of Georgia with specialty in fungal genetics and interspecies variation. Max will talk to us today about revealing your microbes with phenotypic identification. Hi, Max. Take it away.
Thanks, Kasia. As she said, I'm here today to talk to you about phenotypic ID and the different systems we have here at Biolog that can help you monitor contamination and gain insights into your microbial populations. So you've all seen the news articles with various recalls due to microbial contamination, and here we have a few different recent examples with recalls of over-the-counter medication and some common food items recalled because of listeria contamination and other microbial contamination. Cases like this serve to underscore just how critical environmental monitoring is in production facilities. And here at Biolog, we offer a variety of environmental monitoring and identification solutions for you in your manufacturing or research environment. So then what makes for a good environmental monitoring system? Well, we believe that this can be broken down into three major factors. First is ease of use, which is necessary for quick training and reproducible results. Second is flexibility of throughput, as depending on your lab, facility, or research goals, you may need to be running only a few samples or up to hundreds of samples per month. And finally is a comprehensive database, which is critical to success in environmental monitoring, as when you're comparing or trying to identify your species, you want to have a database of relevant and interesting species to compare to, so you are confident in your identification. So combined, these qualities define an ideal in-house system, which can be achieved in a few different ways through the Biolog ecosystem. There are three different levels of throughput offered by Biolog's ID ecosystem, which allows you to choose the solution that is right for you. For lower throughput applications, we have the Microlog M system, which includes our ID software package and relies on the user to enter data manually to be matched against our database. This is best for users running small numbers of samples and can also be used a lot for teaching students the advantages of phenotypic ID and characterization. Alternatively, sampling away from a lab setting where the instrument is inaccessible. The MicroStation is our next option, which is similar to a classic plate reader, where plates are incubated elsewhere and read one at a time. This system is paired with our ID software and will automatically read and interpret your data to identify aerobic and anaerobic, as well as yeast and filamentous fungi. The MicroStation is best for facilities wishing to test smaller batches of samples and do not require incubation and reading. Finally, our Odin system is the all-in-one solution for microbial identification. This platform is able to incubate and read up to 50 plates at a time, containing any combination of aerobes, anaerobes, yeast, and other fungi. Odin will read plates automatically every 20 minutes and call an ID as soon as it can, reducing the time to ID compared to the Microlog manual or MicroStation systems. So any identification system is only as good as the database. So what type of coverage do we offer? What kind of organisms can you ID? How does it call these IDs? All these things are very important. So for Biolog, we have a comprehensive phenotypic database, which covers roughly 1,500 aerobic taxa, over 350 anaerobic taxa, 260 yeast species, and 710 filamentous fungal species. In addition, if your species is not listed in our current database, you have the ability to also generate a custom ID database as a user. And we also offer the ability to ID not only species, but also strains. So if you have different clinical isolates of the same species, we can oftentimes parse those as well. So this all works by using our phenotypic ID system that takes advantage of 95 different phenotypic tests. And so with more tests comparatively to other systems out there that use 20 to 48 tests, we have higher resolving power and the ability to more accurately identify these species. We also have a simplified test procedure. So we eliminate the need for any more consumables outside of just the inoculating fluid and the plate. There's no gram staining necessary, no imaging necessary, no pre or post tests, no DNA extraction. All you have to do is make your cell suspension and add it to the plate, and you're off to the races. It takes just about a minute. So each of our identification systems utilize the same metabolic fingerprinting technology. So here we have an example of one of our microplates, and each well contains a different nutritional substrate or stress inducer, in addition to our universal redox reporter dye, which irreversibly turns purple in response to metabolic activity within the cells. The dye acts as a terminal electron receptor and is reduced by NADH and NADPH to change color. We can measure this color development and use the pattern of wells as a fingerprint, which can be matched against our database, either manually using MLM or automatically with MicroStation and Odin. So now I'm going to dive in a little bit more into each of these ID systems. So the manual system, samples are incubated offline and you read by eye using presence or absence of color development as your output. And using this system, you're able to identify around a little over 1,500 different aerobic bacterial species. And again, this is good for labs which are using low volumes or educating. Next, we have the MicroStation ID system. And again, here, samples are incubated offline and then placed in the reader one at a time to be read. And this is able to identify everything in our database from our aerobes to anaerobes, yeast, and fungi, and filamentous fungi. And so this is best for lower throughput applications where you already have an incubator, but just are doing endpoint reads at an appropriate time, depending on the organism, anywhere between 6 and 22 hours. Then finally, we have Odin, which is, like I said, our all-in-one solution. So we have the simple experimental setup that allows you to identify any type of microbe in our database, and these can be side by side within the instrument incubating at the same time. As long as they take the same temperature, you can be running multiple batches and multiple experiments all at once. So this allows you to test a myriad of different samples all in the same platform. So now it's time to take a deeper dive into each of the ID assays specifically and how they work. So first, our Gen 3 microplate is our comprehensive aerobic bacterial identification solution. This plate was created by screening through nearly 2,000 phenotypic tests to determine which ones were the most useful for parsing microbial identity. Additionally, without the need for special pretests like gram staining, users are able to rapidly move through samples and inoculate many plates in a single workday. Further simplifying the process is the low cell density required, where even a single colony can be sufficient to identify successfully. So here are a few real-world examples of what these Gen 3 plates look like once they're incubated. So we have 4 different species here: M. cataralis, C. pseudodiphtheriticum, Streptococcus pneumoniae, and Acinetobacter baumannii. And each of these have a very different phenotypic profile depending on their different metabolic requirements as well as their sensitivities. So we can see here that we have a negative control in each plate that should not have any color development, a positive control that contains glucose, where each of the organisms should grow, and then we can see that in the right-hand side of the plate where our antibiotic resistance is, that Acinetobacter is actually one of the only ones that's resistant to all those drugs. So not only can you identify your organism using these plates, but you can also learn more about it as well. And as I've been mentioning a few times now, the setup protocol for this is very, very simple. You simply need to have a purified culture, and you pick one colony or a couple, and then dunk it into your solution to make a cell suspension, and then inoculate it into the plate and incubate. And that's it. It only takes about a minute. Now I wanted to take a break from talking about the plates and to go into the software a little bit and to show you what you could expect using the Odin instrument to identify organisms. So this is what you're greeted with in the software. We have our batch creation and loaded plates here. You can see we have multiple samples running at the same time. We can give you an estimate of how much time is left in the incubation, as well as live views of what the growth kinetics and metabolic kinetics are doing at that time. If you want to take a more fine-grained look at your data for data curation, you can do that and look at and make sure that not only are your results reliable, but also reproducible by making sure that your negative controls look good and that your run-to-run variability is low. And we also offer the ability to export your data as a CSV or JSON file. And finally, through the software, you're able to generate a report that is both clear and informative and also compliant with 21 CFR Part 11 regulations. So here we have a few different pieces of information about the user, what the sample was loaded, how it was incubated, where it was incubated. Any user-defined information, such as sampling location, will also be recorded here. We have areas for signatures, if that is required by your organization. And we have the results down here and how long it actually took this organism to ID. In this instance, it was a template, so it ID'd in 14 minutes. But typically, identifications take anywhere between 4 to 12 hours for most aerobic bacteria. And the next piece of information on the report is the table of results, where the top one here is going to be the actual ID of the organism, followed by the next most likely matches. And then we have at the bottom here a heat map showing the relative color development at the time of ID for both the 590 wavelength, which is the dye, as well as 740 wavelength, which is a background control for biomass. This also indicates any false positives or false negatives on the report as well, relative to what the algorithm was expecting. As I mentioned, we also offer the ability to have 21 CFR Part 11 compliance as part of the software, and part of that package is being able to generate and export an audit trail. And so if you are interested in those features, please do not hesitate to ask us for more information about that. So back into the identification plates. So now we have the anaerobic identification, or AN microplate. So this, again, contains those 96 unique metabolic tests, and you can use either gram-negative or gram-positive species here. And again, no gram staining is required. And the big difference for the anaerobe plate is that it is just a single read at the end of 24 hours of incubation. This is because of the aerotolerance of the strains. You need to seal the plate in an anaerobic chamber or box, and then incubate for 22 hours and then read once, either in Odin or microstation, or the Odin or microstation. So next we'll talk about fungi. Fungi are diverse and also very complicated, as many of you, I'm sure, are well aware. So the first question that most people will ask about a fungus whenever they are confronted with an unknown, is it a yeast or a mold, or is it both? And typically, we divide things up into yeast and filamentous fungi based on colony morphology, on our Biolog universal yeast media or a malt extract media. And so if it's filamentous on either of those, it'll be classified as a filamentous fungus for the sake of our identification methods. So to talk about the YT plate, or yeast plate, again, this has 95 different carbon sources used by different fungi. But we split this plate up between oxidation and assimilation tests. So only half of the plate has dye, the top 3 rows here indicated in purple. And then the bottom half of the plate is using OD 740 instead of the OD 590 to measure turbidity or cell proliferation in those wells. And this protocol is very straightforward as well, although it does take a little bit longer incubation-wise compared to the Gen 3 plate, as yeasts tend to grow a little slower than bacteria. We typically ID these organisms in around 48 to 72 hours. Now for the FF plate, we have 710 taxa in this. There's a lot of different environmental, soil, and health relevant organisms in this database. And again, it uses the same redox dye technology, but it's a slightly different dye that absorbs at a different wavelength. This one is 490 nanometers, is more optimized for filamentous fungi, but it works on the same basic premise of metabolic activity as the output. And this protocol works for both sporulating and non-sporulating filamentous fungi, so you don't have to worry about whether or not you can actually gather spores. It's simply you need to macerate the mycelia after taking them off of the plate in order to get an even cell suspension to inoculate into the microplate. And this protocol takes a little bit longer, again, because the filamentous fungi are a bit slower growing. And so we typically see IDs here between 48 to 96 hours, with most of them being in the 72-hour time area. We also offer the ability to verify identification of filamentous fungi by comparing to macro and microscopic images in our database using the MicroStation. So what that looks like is when you get your report for your filamentous fungus in the Microlog software, it will give you both what your color development data look like, the turbidity data, as well as your ID and the next top choices. And you can look for more information of this top ID choice and see that this is the morphology of both the microscopic and the macroscopic. And you can look at that for both the first and second options. And this allows you to not only have a little more confidence in your result, but also keep track of your strains as you culture them in lab and just do quality control. So just to kind of summarize the different varieties of identification plates, again, we have the Gen 3 plate, which is designed for aerobic bacteria. And we have 3 different protocols for this. However, most organisms will fall under protocol A. And anything that you want to identify with Gen 3 plates typically requires to be grown on our bug plus blood auger in order to get it into that metabolic state that we need it to be in. And you can read these plates on any of our identification systems, the manual system, MicroStation, or on our older instrument, Omnilog, or Odin, our new instrument. Then the AN microplate is for anaerobes. Again, this is blind to gram stain, so you can use both gram positive and gram negative on this. And this can be read on our MicroStation or Odin. And then YT, which is for yeast, and you culture things on our Biolog universal yeast media. Again, can be used on MicroStation or Odin. And finally, FF microplate. We use 2% malt extract as our culture media, and then it can be read on MicroStation or Odin. Just to give you a little bit of confidence in our ability to identify these organisms, we have a pretty long history of within the industry and have thousands of customer publications out there worldwide in a myriad of different market sectors, including microbial QC, environmental monitoring, agriculture, ecology, basic research, everything you can think of. And here are just a smattering of our ID customers, including companies like Bayer, Alcon, SeaWorld. So there's all sorts of applications for this ID technology. Before I leave you, again, these are our 3 systems, Microlog M, MicroStation, and Odin. And I implore you to see which system is best for you and your needs.
All right, and with we are running some end of the quarter specials now, so you're going to need an identification plate to whatever you're trying to identify on our systems. So please contact your sales representative or email us at info@biolog.com.
Well, thank you so much for your time, and please do not hesitate to submit any questions to the chat.